Well, thank you very much for the invitation, and thanks organizing committee for putting everything together. That's a lot of work. Also, I would like to thank Dr. Shankar for that excellent talk, because building on that scheme, I think it's a little easier for me to explain the project we are conducting in the lab, which is a translational research on a gene called androgen receptor. Potentially can serve as a novel targeting target in treating glioblastoma or GBM. Here's my disclosure and just a quick introduction. GBM, as you all know, mostly, it's a most aggressive type of glioma and the most common type of primary adult malignancy. In the Central Brain Tumor Registry of United States database, we can find glioblastoma actually accounts for about 15% of all primary brain tumor taken together, and about half of the primary malignant adult tumor. This tumor is hard to be resected completely, mostly due to the large size, that's the initial diagnosis, the critical location, the infiltrative pattern. So the standard care for this tumor right now is based on maximum safe resection of the tumor, followed by 6 degrees of radiation, which is 6 weeks of long course radiation, followed by with concurrent and adjuvant temozolomide, an oral form of the chemo. Although nowadays the NCCN guideline added the tumor treatment field into the modality, but is out of the scope of the talk today. And the benefit of adding chemo has been demonstrated by a STUP trial, which is a phase 3 study. Even with this aggressive treatment, combining surgery, radiation with high dose and temozolomide, we still see very dismal prognosis of this tumor, with rarely long-term survivors beyond 5 years, as you can see here. And even you add the temozolomide to the radiation treatment, in the overall population, we only see a median overall survival benefit of additional 2 to 3 months. So it's a challenging tumor to treat. And in the recurrent situation, the treatment option is very limited. Most of the treatment is based on bevacizumab in combination, as you heard from Dr. Shankar earlier. There's a RTOG 1205 randomized phase 2 study compared adding re-radiation to the progressive tumor on top of the bevacizumab. And in that study, with the result just presented recently, the bevacizumab alone group only achieved a 6 months progression-free survival rate of 29%. By adding the re-RT to the tumor with 35 gray in 10 fractions, we can double that number. But overall, you can see most of the people will progress within 6 months. So the mutation pattern of GBM is pretty heterogeneous. We can find very frequent mutation or tumor or gene amplification in a gene called EGFR or epidermal growth factor receptor in about half of the patient. Combining the PTEN mutation, deletion, and the other gene in the AKT pathway, we see those mutations in about another 50% of the patient. The other cancer driver genes, such as HER2, PDGF receptor, MET, has been reported, but in much less frequency. One of the reasons we think this tumor is difficult to treat is it contains higher proportion of cancer stem cells or glioma-initiating cells. Those cells, they are very resistant to conventional treatment, such as radiation or chemo. They survive, most likely after treatment, and repopulate to grow the tumor afterwards. And another reason, as Dr. Shankar discussed, is the very high intrinsic immune suppressive feature of the tumor, which prevents the immune system of the patient to recognize and attack the tumor after conventional treatment. So in our lab, we're trying to work on a gene called androgen receptor to try to attack both of the issues. As you know, some of you may know, androgen receptor has been used as a target for a long time in prostate cancer patients. Why androgen receptor in GBM? So we did observe a gender difference in terms of GBM incidence with male presented with 1.5 to 2 times higher incidence developing GBM compared to females. And also in the previous animal study, if we implant the GBM to the female mice, they grow slower. A lot of labs, quite a few labs also reported overexpression of androgen receptor in the GBM tissue in human patients. And one of the important reasons is androgen receptor has been studied for so many decades in prostate cancer patients with many available drugs that can be repurposed into GBM treatment. Androgen receptor is the cytosol hormonal receptor for testosterone or dihydrotestosterone. Once bound by the testosterone, it can dimerize, enter the nucleus, binding the DNA, and interacting with those co-regulating factors to drive the downstream gene expression. This nucleation and binding of DNA can be blocked by those drugs we use in prostate cancer patients. For example, bicalutamide, enzalutamide, apalutamide. And all three has been shown to have CNS penetration capability. So using those drugs with very known toxicity profile and brain penetration, hopefully we can develop some study on the GBM. We connected a large number of GBM patient sample from our own institutes and stained the specimen with androgen receptor using the immunohistochemistry method. We see the overexpression of AR in both primary diagnosed GBM and recurrent GBM as well. And comparing to the normal brain autopsy sample, which shows very low or non-expression of the AR. We did once in a while observe very low expression level of AR in the patient with epilepsy status post temporal lobectomy, but in very small number of cells. And in the other 58 patients we looked in our institute, if we define the positive ratio of being 1% or higher for AR expression in all the GBM tumor cells, we see majority of the male and female patient being in that category. And if we define by 10%, we still have 80% male and 2/3 of the female have AR overexpression. And this just shows you some patient on the left with majority of the tumor cells being AR positive, showing this in brown color, and another patient with less than 50% positivity, and another rare case with almost none of the tumor cells being positive. So the next question is whether the overexpression of AR has clinical significance. We look at the tumor cancer genome database. In the MRI expression level, we see the expression level of AR seems to correlate with the grade of the glioma. GBM being the highest grade has the highest expression level of AR comparing to low-grade glioma. And low-grade glioma in turn has higher expression level than the normal brain tissue. And in low-grade glioma, we did see the prognostic value of this gene expression. For patients with high level of AR expression, they have much worse median overall survival compared to the AR low expression patient. And the TCGA is a database mainly containing Caucasian patients. Interestingly, when we look at the Chinese glioma genome database, we also see that the AR expression level seems to be prognostic. For example, in the low-grade glioma patient, we know those patients with IDH1 mutation and 1p90q co-deletion, those people tend to have the best survival. And those patients has the lowest AR expression level compared to other subgroups. The same for GBM. Yeah, we know IDH1 mutant GBM patient has the best survival. And those patients has lower expression level compared to the IDH mutant wild type patient. So it does seem AR expression level has prognostic values in glioma. However, when we look at the GBM patient population only, this prognostic value doesn't seem to be significant. We're still investigating on this, but I think the explanation might be the survival of those GBM patients are so poor. So the prognostic value of the AR expression probably are lost or very minimum between the expression, different subgroups. We saw that in the, based on the mRNA level on the TCGA database and on the protein level from our own patient population. So the next question is whether the AR expression level has any predictive value to androgen receptor antagonists or suppression. So we look at those commercial available GBM cell lines in vitro, in cell culture. This MGPP3 is a mice, it's a mouse GBM cell line, all the rest are human. We treated those cells with increasing concentration of enzalutamide, which is showing on the top, and bicalutamide on the bottom. And we can see in all the cell lines, we see a significant suppression of the cell proliferation. And in all cell lines, the IC50, or 50% of suppression concentration of the drug, is about 30 to 40 micromolar. Very similar, seems to be independent of the AR expression level in those cells. This IC50 is slightly higher than the physiological serum concentration that we can achieve in the blood, in the human blood, which is about 20 micromolar. But again, both drugs seems to work pretty well. And after drug treatment, the AR protein expression shows the dose-dependent decrease along with time. Sorry. And we further look at the c-Myc gene expression in the tumor cells by increased concentration of enzalutamide treatment. You can see the dose-dependent suppression of the c-Myc as well as the AR suppression. c-Myc is an important oncogene that's important in tumor cell proliferation and for cancer stem cell stemness maintenance. And indeed, we performed further study looking at the relationship of the AR suppression and the cancer stem cell maintenance. And in this tumor spheroid study, we see the suppression of the tumor sphere growth by increasing concentration of the drug, either using enzalutamide or bicalutamide. And our RNA-seq genomic study actually shows many of the cancer stem cell marker gene based on RNA-seq has significant decreased expression. In protein level, some of the marker gene also has dramatic decrease time dependently. Even, for example, OCT4, even the mRNA level is not decreased. In addition to the cancer stem cell gene, we also find those DNA repair enzyme gene also get suppressed by enzalutamide treatment, particularly those involved in the non-homologous end joining process. And when we look at the TCGA database again, we see a very strong correlation, positive correlation of almost all the DNA repair gene expression related to the AR expression level. So it looks like from those in vitro study, the androgen receptor blockade can function as a radiosensitizer. So we proceed into the in vivo study. We established a mice model in the lab by injecting a GBM cell line of the mice, MGPP3, into the one side of the brain by microinjection technique. And those mice can be followed for GBM tumor growth in live mice based on the bioluminescence because there's a luciferase gene knocked into the genome of the mice. We proceed with this tumor model to look at the drug treatment alone, RT alone, or drug plus RT using both enzalutamide and bicalutamide separately. And as you can see, if we just use the drug alone, some of the tumor continue to grow, some can start to shrink and disappear. But if you look at the combined treatment group, either enzalutamide plus RT or bicalutamide plus RT, all the tumors start to shrink and disappear eventually. And it seems the drug and RT combo seems to be tolerated very well by the mice, with most of the weight of the mice kept stable, even some weight gain as the toxicity of the drug. So what we did in this experiment, to be more specific, is we start the drug treatment 1 week before RT. We give whole brain RT 10 gray times 2 once per week. And then drug was continued for about 10 weeks. And mice are followed very long. And with the treatment, we see control mice all died during follow-up. RT only prolonged the survival, but eventually died out. The drug only, either biclopamide or enzalutamide, has about 50% of the mice survived with all tumor disappeared. Surprisingly, if we combine RT and this drug, we see none of the mice died, and 100% of the mice has tumor disappeared after long-term follow-up. So we're happy. We think this is definitely proving this group of drug can be radiosensitizers, most likely by suppressing the tumor cancer stem cells or glioma initiating cells. And we confirmed that in the in vivo study by IHC or immunohistochemistry. What surprised us is this rechallenging study. So we kept the mice survive for a long time and rechallenged those mice after the tumor disappeared by injecting the tumor cells to the contralateral side of the brain. And surprisingly, we only see one mice develops new tumor out of a total of 8. And that only mice developing tumor is from the biclopamide only treatment group. All the 5 mice from combined treatment never developed new tumor. So this indicates there must be some kind of immune memory developed for those tumor in those mice that survived combo treatment. And indeed, we proceed to look at the in vivo tumor tissue. And we find that enzalutamide alone can significantly reduce the regulatory T cells. And as you heard from Dr. Shankar, it's a factor that it's a cell type really maintain the status quo or function as immunosuppressive in the tumor microenvironment. But combining with RT, not just the drug alone, we also see significantly improved infiltrating of the cytotoxic CD8 T cells and CD4 cells. We look at the genomic response of the drug treatment as well with RNA-seq study. And we find that this consistent pattern of drug suppressing the immunosuppressive genes in the tumor microenvironment, particularly all the genes involved in the STAT family, as well as IL-6, IL-10, LIF receptor, and some of the factors you saw on the slides from Dr. Shankar's talk. So I'm going to skip this one. So just briefly, we look at the Western blot's result investigating on the signal transduction pathway. We find consistently STAT3 phosphorylation pattern are inhibited or altered by this drug treatment. And nowadays we know that STAT3 phosphorylation is downstream of many growth factors and cytokines, which are important for the promoting the GBM. So hopefully, by studying the relationship between AR and the STAT3, we can find a niche trying to target the stem cell and the immunosuppression for this tumor at the same time. So in summary, we do see a radiosensitizing effect of AR antagonists combined with RAN RT. Initially, we thought it's a suppressing of stemness of the tumor cells, but nowadays we think it's probably more, or at least in additionally to what we saw before, immunomodulation of the tumor microenvironment. So from all the study, preclinically, now we're developing a phase 1 clinical trial, trying to translate those study results into clinic, try to help the checkpoint inhibitor that has failed multiple times in treating GBM. And we saw in animal study that drug alone doesn't seem to control the tumor completely, indicating there's insufficient dose in the brain. So we're going to start with the 3x3 design for dose escalation for biclopamide to start with, to be combined with brain RT using the RTOG 1205 protocol. And we can talk more in details if anyone are interested and collaborate. So I'd like to thank my mentors and collaborators for moving the projects. Thank you.